Hello, I'm David Wood. I'm Program Manager for the Living Murray at Mallee CMA, and prior to CMA I worked in fisheries research and science along with this man, so whatever I'm talking about science-wise I learnt it off him, so if I get it wrong he won't care, doesn't matter. Alright, so yeah, we're gonna have a quick look into fisheries research and how that can be used to inform management. Alright. I don't think you can have effective management without good science backing it up. We're just making it up otherwise, and that's not what we want to do. As water managers, what we're trying to do is give nature a hand. You've seen that flows aren't what they used to be. We're trying to fill those gaps. We can't perhaps do it on the big scale, but what we're trying to do is just work on little scales in the first instance. I work for Mallee, that's the scale I'm working at. We can't deliver water everywhere all across the Mallee to every wetland every year. It's just not possible. So we're working on just little locations. When we plan for waterings, there's heaps and heaps of considerations. Number one in pretty much every watering we do is environment's number one. And there's a whole suite of things we can work at there, whether it's fish, birds, vegetation. So having really clear targets and goals is really, really important. Other considerations are definitely community, social, economic, and also cultural values that come into waterings. Ideally, science should inform all these management decisions. We try and use the best information available. Sometimes we don't have really specific information. We can just use, we know golden perch like large flows. That's the information we have. But sometimes in some locations we have really good knowledge of what makes that system tick or that species tick. So that's the information that we're trying to use. And it's all about adaptive management. Keep asking questions, review what's gone before and what information's coming in now. Apply that newer information where you can to your decisions and what you want to do, and keep testing the outcome. Make sure you're getting the result that you're aiming for in the first instance. So, a little bit on the research side of things. This is my old background, if you like. But first you need to catch the fish, and there's a whole bunch of different methods you can do that. Obviously netting and different nets and sizes and types depending on what you want to catch. Fish weight traps. Angling certainly used in the science world. Some of those species are fairly finicky to try and gather. This guy down here is a light trap. That'll catch larval fish. You put a glow stick in that, set it out at night and it attracts the little fish. And of course electrofishing. And one of the questions I got very frequently, how does electrofishing work? So I'll try and run you through it really quickly. So on a boat, electrofishing, you need power, electricity. So that's generated by a generator on board the boat. That power goes to this little thing here, it's a control box, and that allows us to manipulate the power and turn it into a form that we can use to push into the water. So for those that have some electrical understanding, we're using direct current normally, and that's pulsed into the water. All right, other parts of the circuit, back to primary school electronics, you need a positive and negative terminal if you like. For the sake of electrofishing, the negative terminal is the boat hull itself, and then these guys out the front are called the anodes. They're the positive part of the terminal. And what connects that positive to the negative is the water. And fish in that water, in that effective field, they're essentially stunned. They come to the top and they're netted and put into the boat. So the other question is, why aren't the guys on the boat electrocuted? It's because they're at the same potential as the boat. So think about a bird sitting on a power line. They're not getting electrocuted because their body's at the same, let's say, power as the line that they're standing on. It's when you go and touch something at a different electrical potential, the energy wants to flow from one to the other, that's when there's problems. So, of course, when you backpack electrofishing, you're standing in your own electrical field and it just seems ridiculous. But luckily, as long as your waders don't have a hole and you're not touching the water, you're good. All right, so research, what data and information can we collect? I'm saying we, I was a scientist. I like to think I still am. So larval fish, they're really small, millimetres, and eggs. We can identify them. There's guides to say that, you know, there's certain features that make it this particular fish. It's obviously not as easy as when they're fully grown and adults. And also there's physical traits on the fish that tell us how old they are. You know, there's certain morphological traits that come through, you know, every couple of days. Really simple things, measure and weigh the fish. That gives us a whole bunch of really useful data that's easy to collect. DNA samples, population studies, that sort of thing. Otoliths, so those ear bones. As Ian said, you can age them just like trees on a ring. And micro... Rings on a tree? Everyone knew what I was talking about. It was only you that was heckling. All right, so, and then microchemistry. So that's what Ian was also talking about, where there's particular chemical signatures in different waters, and when you grind down the ear bone and certain parts of the ear bone and put it through a process, it tells you what the signature is, and you can match it up and say whether it's from the darling or the bidgie or somewhere different. Whole bunch of different tag types. Dart tags, these guys, most common to anglers. They're on the outside of the fish. They'll have a name, they'll have a phone number. If you catch that fish, call it in. From that we can tell how far the fish has moved, how much it's grown across that time. Pit tags, so these guys, they mostly have recorders on all the fishways up and down the river. That tells us when fish are moving, which direction they're moving, except for Lock 10 obviously. 11. Radio and acoustic transmitters are also tracking devices. They're surgically implanted into the fish generally, and they release a signal every, 20 seconds, 30 seconds, and there's listening stations all around the river that you can put out. And as the fish swims past, it picks up that signal, and you can determine when and which direction that fish has moved past. So that gives us really good understanding of where fish are moving and how, and does that relate to a flow or a particular action that you've undertaken. There's also stuff like controlled lab experiments, such as swimming speed, which is really important to know for the likes of when you're setting up a fishway, and environmental tolerances, so how salty can the water be before the fish doesn't breed or the eggs don't survive when they're spawned. So managers, what do we do with that information? We can support fish movement. We then start understanding what flows might trigger a particular response, and we can aim to target them when we're delivering water. We can provide habitat, so hydrological, whether that's flowing velocity or just a particular peak in flow at a certain time. Structural, are we inundating a whole bunch of habitat by releasing more water somewhere, or do we need to re-snag a site? And then also fish-like habitat. Some of those small guys, they like aquatic vegetation, so we may not be targeting the fish per se, but we're trying to create lots of habitat for them through the growth of vegetation. Provide those nursery habitats, connect to wetlands, let the fish grow up in the wetlands, and then let them get back into the Murray through manipulation of regulators, for example. And we can identify and remove barriers to fish movement and also control pest species. So a little bit of a case study on what's happening out at Mullaroo. Many of you are probably aware of it. It's out on Lindsay Island, part of Murray Sunset National Park, over near the South Australian border. And the creek itself splits away from the Murray just above Lock 7 and flows down into the Lindsay River. The gradient along that creek line is fairly steep in comparison to a lot of the other places around here, and that creates really good fast flowing habitat, so the water velocity is quite high in that site. It's also full of snags. Think of it as what the Murray would have been like 200, 300, 400 years ago. That's what it's replicating, and all that means it's perfect for Murray cod. They really love this site, and there's a really strong population going on there. So, lots of science being undertaken on this site. It is important, so we keep looking into it and make sure we're doing the right thing by the fish there. Some of the stuff that we've looked into, So velocity profile, so how fast the water is flowing and whereabouts. So that's what we're looking at up over there. What we're looking for is that orange and red dots. That means the water's flowing faster. So the top one, the flow is lower than the bottom one. With higher flow, we get higher velocity across a larger proportion of the creek, which is good for cod. There's snag density mapping's been undertaken there. Just by measuring fish, we get some really cool information here about length frequency and what the population looks like of this fish, of this species. This is data from 2018, 19, 20. See here, not many fish caught generally, and what was caught small. What's really cool is lots of these small fish have grown up in the last year, have been spawned in the last year. And over these 3 years, there was new fish spawned every year. The other thing that's cool is these fish are surviving. They're now this age class structure, and then as we get to the next year, they're growing bigger again. So we're starting to bring back a really solid population structure. There's not a lot of big fish, but you know, there's some fish coming up to fill that gap, which is really cool. Larval sampling will tell us if the fish are spawning in the first place and what sort of time frame they're spawning in. So is it, you know, early spring, late spring? And that gives us lots of information. Movement patterns, so through a lot of the telemetry, radio tags, acoustic tags, it gives us information like this. So this one, across a particular time frame, it's showing that smaller fish don't generally want to move in the Mullaroo, while the larger fish are more happy to move. This other one, essentially what it means is it's showing fish movement through the regulator upstream, moving upstream, and during spring, they're more likely to move than they are during winter. We've also undertaken fishway sampling to find out the effectiveness of the fishway. So for management, we're using that information, we're using it to support a strong population year round. We know what thresholds that the fish do and don't like, and we're trying to manage the creek line within them. One thing that we have played with, and starting about that 2018 where we started seeing some strong recruitment, a higher flow in spring is really good for the population in the Mullaroo. It's supporting them breeding, it's providing lots of habitat, both through that flow and velocity, higher flow, but also it inundates more snags. Push more water down a creek, the height increases. It also supports their movement through the fishway and up and down the creek itself. We don't stop there, we keep analysing and looking at what the fish are doing just to make sure we're doing the right thing. It's no good doing the test and going, yep, we're good, it's all working, off you go. You've got to keep checking and make sure that it's doing the right thing and keep adjusting what you're doing to make sure you're getting the best outcomes. It's also good to improve that working relationship between the scientists and the managers. As I said earlier, the science is where it's at. That's what's informing what we're doing. And as I said right up the start, I'm doing what I can on my little patch, but for many of these species that anglers target, they work over such a broad landscape. Golden and silver perch move thousands and thousands of kilometres. I can't make them breed on my little patch of land. That's not how they work. So working together with other states, water managers, basin-wide, that's where it really should be at for management of fish, particularly those large-bodied angling species. There are still instances, though, where management of local populations is important. For example, threatened and endangered fish and populations, like at the Mullaroo. Unfortunately, it's not all about fish. We are here talking about fish, but as a water manager, I need to consider all other aspects of the environment. You know, as I said, birds, vegetation, all that sort of stuff. So fish are important, yes, but they're just one part and we've got to manage it so everything benefits. And that's it.